Very good day. I'm Dr. Ashok Rathor. Here we'll discuss about stability studies of drug products and including good warehouse practices and receiving of material and dispatch of the product under the GMP. So here you can see before going to discuss about the stability of drug product, we have to understand what are the drug products. So here in this slide, you can see there are many drug products available in the form of injectable, sometimes ointments are there, syrups are there, tablets are there, capsules are there. So there are number of forms, we can say different type of drug products are available. So these drug products are the finished product of any drug which is available in the market, and these drug products are ready to use by the end user or the customers. So the drug substance, because they have a multiple factor and because they are mixed with some component, because this drug should be reached to the end user in the original form, and due to the transportation, due to the storage, and these because this API means active pharmaceutical ingredient, which is contained by some type of recipient, and these are sensitive. So some other components are required to carry with, to mix with, to carry to the end user before releasing into the market. So the drug substance is active pharmaceutical ingredient, or simply we can say API, which is sold in the market, and it can be a pure which exert pharmacological action on the body. So these different type of drug products available in the market. So what type of medicine or what type of drug products are available? You can see first in the form of liquid. That can be said as a syrup. So active part of this medicine combined with the liquid to make easier to take, or simply we can say to absorb in better way by the specific target. Second one is the tablet. Means API is combined with another substance and pressed into a solid form, round, oval, or some different shapes are there, but solid form. That is known as tablet. Capsule is again a solid form. Here the API, or we can say active part of medicine, is contained inside a plastic or gelatin cell that dissolves slowly in the stomach. Especially some topical medicines are there, like they are ointment, lotions, cream that can be applied directly on skin. They come into tubes, like in bottles, sometimes depending on the type of medicine which you are going to use. So active part of medicine mixed with another substance, making easy to apply on the skin by the end user, by the customers, by the users. Some type medicines are coming in form of suppositories. Here the API is combined with another substance and pressed into a bullet form. So it can be inserted into the bottom, and these suppository must not be solid. Sometimes you can see the drops are coming from for use and use, like in the eyes, ears, nose. So these are known as drops, and these medicines are best works where they reaches to affected area directly. So for that reason only drops are there. Sometimes you can see the inhalers, to be used in asthma or some other type of diseases. So these should be, these medicines are API of the medicine released under the pressure directly into the lungs. Another form of medicine are tablets, sorry, drug product are injectable form. Of course, they are in liquid, but liquid we cannot put under liquid because they are injectable medicines, injectable drug product. So here, two type of injectables are there: subcutaneous and intramuscular. So depending on where it is going to take, using the injection in the muscles, in the veins, etc. So, and the last the product is implants, or we can say the patches. So these medicines are absorbed through the skin, like nicotine patches, which help giving up smoking. Sometimes these implants are available uh, today to use by the end users in the market. So here you can see the tablet in the capsule. The different type of tablets are there. So shape can be depending on this, because these are not having any type of legal type provision, but this can be made by the company for their we can say marketing. But shape and size can be also depending on the factors, stability factors, so on which they have, because during the transportation, during the warehousing, I mean storage, and sometimes distribution again, it requires from dispensary to the local, we can say local warehouse, the dispensaries, then it come to the end user. So during this transportation and storage, means some factors are there, climatic factors are there, so they may lose their potency. To retain their potency as it is, whatever is it is manufactured. So after many studies, they have, means R&D section will decide the shape, size, and the component which can be mixed with the API. Here, before going to discuss about the stability study, we have some equipment here. You can see, like start from analytical balance, autoclave, auto titrator, bacteriological incubator, BOD incubator, calculator, scientific, many things are there. These are the list, number of list of available, which can be used in manufacturing of medicines. Okay, and especially for tablet manufacturing, if you see here, like blister machine, bunker, case blender, clint mill, coating machine, collide mill, a compression machine, fluid bed dryer, leak test apparatus, many things are there. Many, and for liquid manufacturing, a syrup homogenizer is available, inspection system, labeling machine, low pump, packing conveyor, rope cap loader, many things. Jacketed vessels are required as per capacity 125, 250 liters or 500 liters as per the requirement by the manufacturers. So what is the stability exactly? Stability means, if you say by generic meaning of stability, means how long it can contain, it can maintain its original form, whatever the API it has. So the stability of pharmaceutical product means the time period, how much time period they have to maintain their original form without changing any visible changes, or we can say without any visible changes under the influence of environmental factor or climatic factors like temperature, humidity, light. So we have to consider all those factors. If there is no change, that is the shelf life. And for the shelf life, to fix the expiry date, to fix the shelf life, we have to go for stability studies. So stability study of pharmaceutical product are one of very important parameter for even for R&D are developing a new drug. So this because we need to check, we need to fix the shelf life, because this shelf life play a major role for pharmaceutical product and also its development of new drug in the dosage form, to determine the particular storage condition and to label instruction as per GMP. Our US FDA, many means licensing instructions are also there. So as per statutory requirement to ensure the maintenance of product quality, safety, efficacy throughout the shelf life, they should be considered on the acceptable criteria. They should be approved for pharmaceutical product. So this is required. Stability study is required. Here, in this section, we'll discuss about two things: tablets and capsules. So we must understand what are the exactly tablets and capsule. Tablet is solid dosage form, easily obtained by a single or multiple compression. As I said, these are the solid form that can be means compressed under some producer in the form of tablet. Some different shapes are there. You can see a picture on the slide. Compression of powder, sometimes means granules. This can be said as a formulation, and after compression, will get some type of shape. And these, in some certain cases, tablet may be obtained by molding as extrusion technique. So they can be two type of tablets are there, coated and uncoated. So and normally right surface solid cylinders, they have end surfaces which are flat, or sometimes they have convex flat edge, or sometimes they have beveled shape on the edges. So sometimes some tablet have a break mark, that is known as scoring, or some symbol, some other marking like in the form of dosage for marking into 2 part or 4 part. Capsule, these are medication that enclosed in an outer shell, and this can be made of gelatin or some we can a container of gelatin, a cell where the drug should be there, API should be there. So the outer cell is broken down in the digestive tract, and the medication is absorbed into the bloodstream. After absorption in the bloodstream, this can be distributed, metabolized in our system, in our digestive system. Capsules are easier to swallow and used by manufacturers when the drug cannot be compacted into the solid tablet. Capsules are made only when this cannot be converted into the tablet, because in the tablet form, it is easy to have the shelf life, or we can say the in case of tablet, they have more means longer period of expiry time or shelf life. But in case of capsule, they have they can be broken down, they can change some time after some time and during transportation, so they have less shelf life. So what is the purpose to study the stability? So first, as I said, to fix the self-life determination. And because under the temperature, humidity, some light intensity, we need to store these product. So self-life determination is important during means by which, from which we have to decide the expiry date. Stability testing is required as per some statutory requirement, because every pharmaceutical product should be stored at a normal, and sometimes we have to store at accelerated condition without degradation. So this is required under statutory condition requirement also. And storage condition recommendation, to fix the storage condition, like in freezer we need to store, or in the ambient temperature we need to store, or what type of storage condition for the labeling. So we need to fix all those storage condition of the tablet, of the pharmaceutical product. Elimination of impurities, because during the study we need to understand about the impurity, or because the acceptable criteria. So this can be analyzed during stability study only. So if there is any impurity, then we can go for the elimination of impurities. Then also this stability study required for new development, new product development. So this is again effective for R&D professional to redesign the existing product and to develop a new product. This also requires to ensure the quality, because under the pharmaceutical or US FDA, QA/QC, quality assurance and quality control is the most important attribute when this can be before going to release into the market. So as per the QA/QC, stability is required. Also, it is required when we are going for packaging material selection, because during the stability testing, pharmaceutical product is exposed to humidity and temperature. So effect of temperature, effect of what and temperature, some other material, even packaging material, it should be chosen by the stability study only. Ideal container must tolerate in the stress or different type of stress, or the packaging should maintain the quality of the product during the storage as well as the transportation. And some legal approvals are required like ICH or WHO. Then again, we have to carry out the stability studies. So these stability studies primarily cover physical, chemical, and microbiological attributes. So under physical, you can say the first is the appearance, melting point, water content, clarity, color of solution, pH, dissolution, disintegration characteristics, viscosity, crystal modification, and particle size. These are the parameters for physical attribute. Under chemical, the test, that is assay, degradation product, related substance, that is RS, residual solvent, we need to check. Under microbial, the growth of microorganism and efficiency of preservative content, like some as an antioxidant, sometimes we have antimicrobial preservative. So we need to test, we need to have all these parameters under this stability study. How the degradation happens, two type of physical and chemical. In case of physical, loss of volatile constituents contained by the API, loss of water, absorption of water, crystal growth, polymorphism, color changes may happen due to physical degradation. So all these parameters under physical degradation. But in case of chemical decomposition, hydrolysis may happen, oxidation, optical isomerization, epimerization, geometric isomerization, polymerization can happen during transportation, during the storage condition of the API, for absorption of carbon dioxide, and sometimes decarboxylation can happen. Due to these type of reactions, chemical decomposition of that API, that drug product may happen, then this reduce the shelf life. And also the here the influence of light, or how the light intensity can work. So light energy when strike on photolabile drugs, this activate the molecules and this trigger the means reaction. This can enhance the rate of reaction, either type of hydrolysis or isomerization or oxidation or polymerization, decarboxylation, any type of reaction can happen when your drug in question is photolabile. So decomposition due to the absorption of sunlight, particularly in spectral region of visible blue, visible violet or UV. It ranges from up to means we can say 500 to 300 nanometer. So the drug composition testing by sunlight, artificial light, and UV rays, we need to fix that under these type of condition, drug will not decompose. Photochemical reaction usually complex, so this can be proceed in several steps to follow the zero order. Zero order means there is no decomposition on of drug during these type of intensities. And temperature, of course, temperature is also affecting the API. So to calculate the temperature, we are using Arrhenius equation. You must aware about this equation, of course. So speed of many reaction increase about 2-3 folds with every 10 degrees Celsius rise in temperature. So for energy means here we have to calculate the effect or influence of temperature on drug decomposition by using Arrhenius equation. So here is the formula. By using this formula, this equation, we need to calculate what are the temperature, what are the influence of temperature on decomposition of particular drug. So decomposition may sometimes increase rather than decrease. So oxygen content enhance oxidation, of course. Frozen solution can cause some instability or concentration of reaction is more in liquid phase. So it can change the pH, like protein change the isoelectric point, and sometimes this can also change the folding or unfolding reactions of the protein. So all these type of factors, this type of reaction may happen. So main objective of the stability study is to check the environmental condition for the tablet and capsule, to check the stability of drug in time and favorability of drug given time, Whatever we have fixed, we are going to fix. To check the dissolution time in gastric juice, artificial juice, and also we need to check water content of drug in different environmental conditions to determine the release of API in dosage form. And also we need to check the content uniformity, blend uniformity. So these are the objective for today's stability study of drug product. And now we have to understand what are the climatic zones of as per the ICH International Council for Harmonization of Technical Requirement for Pharmaceutical for Human Use. Under means by ICH only, the world has been divided into four main food zone: temperate zone, Mediterranean subtropical zone, hot dry, hot humid, and higher humid. Because this fourth one has been subdivided into two: hot humid and higher humid. Hot humid is also known as tropical, and means higher humid. We can say hot higher humid. So that is zone 4A and 4B. So total zone are four, but four again divided into two, A and B. So here you can see these are the temperature conditions. In case of June 1, temperate, this has 21 degrees Celsius, 40% relative humidity. These are the recommended condition by ICH for long-term stability testing. For zone 2, Mediterranean subtropical, why these are required? Because when we are going to transport our drug in those areas, then we have to fix the shelf life on these bases. We have to check the condition, friability, stabilities on these conditions only. Like you can take example of corona vaccine. If India has prepared the corona vaccine and this vaccine is going to the temperate zone, zone 1, then we have to test the stability of that vaccine, that drug on 21 degrees Celsius, having 45% of relative humidity only. If the particular drug is going to zone 2, then 25 degrees Celsius temperature, 60% of relative humidity, that is subtropical or Mediterranean. And in case of zone 3, where hot humid or hot dry, sorry, not hot, hot dry, then temperature is 30 degrees Celsius recommended and relative humidity 35%. Hot humid like zone 4A, that is 30 degrees Celsius, 65% of relative humidity, and zone 4B have what higher humidity is there, so 30 degrees Celsius with 75% of relative humidity we need to check. So these are the recommended conditions for long-term stability testing by ICH. Here you can see the world map, and in this condition you can see this is the zone 1. Zone 1 has a major area, and in case zone 2 the color is different. Here you can see in the legend, and India is coming on the zone 4 only. So, and zone 3 has very few, means very less area occupied by zone 3, mainly occupied by zone 1. So these are the zones where we have already seen in the previous. And for the stability testing condition, here on the slide you can see is with the temperature variability plus minus 2 degrees Celsius, but in case of refrigerator and frozen, they have variability of 3 to 5 degrees Celsius plus minus with no humidity. And this is the minimum duration, that is it should be for means stability study should be tested for 12 months at least. And these are the means normal condition. But in case of accelerated condition, so testing should be done for the accelerated ambience, accelerated refrigerated, accelerated frozen, and for intermediate with this type of temperature. Like in case of first accelerated ambient, 40 degrees Celsius plus minus 2 degrees Celsius, but temperature humidity is 75 to 75% plus minus 5. And in case of accelerated intermediate stability, it should be tested for 6 months as per this table. These are some equipment where we are going to use. Here one by one you can see what type of equipments we are having. So one is the analytical balance. You must aware about this. Everyone is using analytical balance in the laboratory. Either you are a small laboratory or you have a big laboratory. So these are the balances where we use. Sometimes for moisture balance, we are using IR moisture balance. Bulk density apparatus. These are the blenders. This is the pH meter by which we can have digital pH meter, of course. We need to record the value of pH in decimal point 2. Then we have here you can see one testing, or you can say this determination of pH by digital pH meter. You can just have a look on this.
The instrument seen in the video is pH meter. So the initial setting requires setting of room temperature to 27 degrees Celsius, and the instrument is calibrated for testing of samples to measure pH by calibrating the instrument to 3 different values, 4, 7 and 9.2. For every testing of the sample, the beaker is cleaned thoroughly with distilled water and wiped with tissue paper. The beaker and the apparatus is cleaned with distilled water and wiped with tissue paper thoroughly before the calibration of the instrument is carried out. A sample buffer solution for calibration of the instrument to pH value 4 is taken in the beaker. And the electrode is dipped inside the beaker so that it is placed inside the buffer solution. And then if you can just see, the value is almost nearer to 4, but we can readjust and recalibrate it again. So it's almost nearer to the 4. So the calibration of the instrument to the value of 4 is complete. Every time the instrument is cleaned with distilled water and the electrode is wiped with tissue paper. Then the instrument is ready for next set of calibration. Now the buffer solution for calibration of pH value equal to 7 is taken in the beaker. And then the electrode is immersed into the buffer solution to calibrate the pH value equal to 7. The knob is adjusted so that the pH value is almost equal to 7. Now the calibration is complete. The next set of buffer solution for the calibration of pH value equal to 9.2 is taken in the beaker. And then the electrode is immersed into the buffer solution to make sure the value is almost equal to 9.2. You can adjust the knob to wait for a couple of seconds so that like the pH value is calibrated to value equal to 9.2. So this completes the calibration part. The tap water is taken in a beaker and the electrode is immersed, and the pH value of the tap water is noted from the pH meter. A couple of seconds is needed to be waited, and then the reading is to be noted. The reading is 7.24 approximately. Since the pH value is more than 7, the sample is considered to be alkaline.
So in this video, you have learned that how pH should be calibrated and how we can use the pH meter. This is the disintegration test apparatus. This is the friability test apparatus. Here the dissolution test apparatus. This is for hardness, means to test the hardness of the tablet. Here you can see in the zoom by zooming this, easily you can understand the size of this hardness tester should be fixed as per the size of the tablet. Then on the pressure, hardness should be tested. If there is some material residues are there left behind after working, after completing the test, that means we can record the hardness using this the panel, control panel. Here it is, you can see. And this is the TOC analyzer. This is IR spectrophotometry. Here is a picture of whole spectrophotometry. This is UV visible. You are using this type of equipment.
Let's go over the parts of the spectrophotometer. First, you have the sample compartment. The lid opens and closes to put your sample inside. You have the wavelength control knob to change the wavelength. This is called the zero control on the left-hand side. And this is the transmittance absorbance control on the right-hand side. Right on top is the mode select button. And when you push the mode select button, you'll notice that the LED will change positions. And so will the readout. To begin using the spectrophotometer, you have to have your samples ready. So first, you should prepare a blank solution. And the blank solution has everything in it except for the molecule or compound you wish to measure. Then you should also have a tube which contains a sample which you want to measure. When taking readings, it's best to go ahead and use the transmittance as the way to set up the machine and also to take your readings. The transmittance scale is linear, so it's easy to set the 0 and the 100%. So when you're ready to calibrate the machine, be sure to push the mode button several times until you have the transmittance value highlighted before you begin. In order to start taking a reading, you have to first set the machine up. So to do so, first thing you must do is use the wavelength control knob and adjust the wavelength until the wavelength that you want to measure at is reached. And let's go ahead and try taking a measurement at 420 nanometers. Next thing you need to do is, using the zero control, turn the zero control until you get 0.0 showing up on the display. Next thing you should do is now open up the sample compartment and put the blank solution in. You should always wipe off the bottom of your cuvettes before you insert them in the machine to make sure that nothing interferes with the reading. Also notice that there's a little line shown in the front of the cuvette. Line that up with the dash on the front of the sample compartment and then insert it all the way inside and then close the sample compartment. This time you're going to use the absorbance transmittance control and turn it appropriately one way or the other until you get 100 on the dial. Once you've set the machine at 100%, then you can go ahead and take your cuvette out and then put your sample in. Again, making sure that you line up the mark on the cuvette with the mark on the sample compartment. Here you can see that the transmittance value is 93.0%. You can also take an absorbance reading by simply pushing the mode button to go down to where it says absorbance. So when the absorbance LED is lit, you can see that at the same wavelength, the absorbance is 0.032. You want to ignore that. The machine has to be set up specifically in order to use that option, and this one is not. So do not write this concentration down, and you need to know the correct factor in order to set the concentration. So ignore those. Just use the transmittance and absorbance on the machine. When you remove your sample, the machine should return back to 0 transmittance. You can go ahead and continue to put new samples into the sample compartment and continue to take readings at the same wavelength that you just set the machine up for. So if you have 10 test tubes, you can now put all 10 of them in one at a time and note the readings. But once you decide that you want to record a wavelength at a different wavelength, let's say we want to switch from 420 to 480 nanometers, now we have to go through the entire calibration method one more time. So then we'll have to begin by first changing the wavelength to the desired 480. We'll have to zero the machine using the zero control knob on the left. Then we'll have to put the blank cuvette back in. And then we'll have to use the knob on the right, the absorbance transmittance control, in order to set the machine back at 100% transmittance. And then after that's complete, then you can go ahead and put your sample in to read the absorbance or transmittance at the new wavelength.
This is SPLC. Here you can see another video how to work on SPLC.
Could equally well stand for high pressure liquid chromatography. It is used for separating mixtures, either to analyze the mixture or to separate a required product from others in a reaction mixture. It can also be used to find the relative amounts of different components in a mixture. HPLC works on the same principle as paper chromatography, here shown speeded up. A liquid, called the mobile phase, moves past a solid, the stationary phase. In paper chromatography, this stationary phase consists of water molecules bound to the cellulose in the paper. The mobile phase carries different components of a mixture, called the sample, along with it at different rates. How fast each one moves depends on its relative affinity for the mobile and the stationary phases. For example, if the mobile phase is more polar than the stationary phase, the more polar components of a mixture will tend to move more quickly than the less polar ones. In HPLC, the stationary phase is a solid packed into a column, like one of these. This particular column contains silica particles to which C8 hydrocarbons are attached, making the stationary phase non-polar. In paper chromatography, the solvent moves along the paper by capillary reaction. In HPLC, the liquid is forced through the column by high-pressure pumps. The whole apparatus looks like this. These bottles contain solvents. Two solvents can be mixed in any proportions to give a mixture, the liquid phase, of suitable polarity for the separation that is being done. In this case, one solvent is water, very polar, and the other ethane nitrile, less polar. The operator can decide on a mixture with the correct polarity for the separation she is doing. These are the pumps. They produce a pressure of 15,000 kilopascals, 150 times that of the atmosphere, hence the name high-pressure liquid chromatography. If a single sample is to be run, it is injected into the solvent stream here, in the injection port, via a hypodermic syringe. Alternatively, several samples can be run in succession by loading them into this auto-sampler, which will run them in order without any human intervention. The pumps force the mixed solvents through the column. The solvent emerging from the column and carrying the separated components of the mixture passes into the detector. Here, a beam of ultraviolet light shines through it. This light is set at a wavelength that is absorbed by all the components to be separated. When the detector reading drops, the component that is absorbing UV light is coming out of the column and passing through the detector. Many alternative types of detector are possible. This one measures refractive index. The time that each component takes to come off the column is called its retention time and can be used to help identify it. Here, the HPLC instrument is being used to separate a mixture of two steroids used in pharmaceutical preparation. The column chosen is packed with a nonpolar solid. The tails of the molecules represent hydrocarbon chains C8H17. Having chosen the solvents, detector wavelength and flow rate, a single sample is run by injecting about 20 microliters into the injection port. The more polar component comes off the column first, followed by the less polar. The peak at retention time, 1.5 minutes, represents other ingredients used in formulating the product. This is the pharmaceutical product, and behind it, its chromatogram.
So in this video, you have learned about how to work with HPLC. Another one is the gas chromatography. Here you can see the injectors are there, and the gas chromatography. So now we'll discuss about how the methods, what type of methods are there for stability study. First, we need to go for the assay. In this test, substance present in any drug product are first prepared in a bulk, which may have active, which we can be inactive. Inactive are like whatever the inactive that are known as placebo, which consist of cellulose, as it is also a starch. Some reagent for assay is potassium dihydrogen phosphate, orthophosphoric acid, triethylamine, acid nitrile, and molecular water. To test the blend uniformity, this is important factor in pharmaceutical manufacturing, because poor uniformity of the blend can cause the rejection of the product, and also this can cause a loss of revenue. But there is though no standard method has been developed for this. Here you can see by using this apparatus, one can easily find out the blend uniformity. Like here you can see in this video. This is the blender. Under this, we need to put the tablets like 1 to 10, whatever as per the requirement, as per the STPs, standard testing procedure. We have to fix the temperature, we have to fix the humidity, means what environmental condition as per the climatic zone where your drug is to be carried out, we need to fix here. And after fixing the temperature, the relative humidity, we have to go for the this blend uniformity test. The reagent are the same, like orthophosphoric acid, triethylamine, potassium dihydrogen phosphate, acetonitrile, water. We have to put, and we have to follow the protocol because this is the standard protocol. But because for specific type of drug, the specific test procedure is there. To test for the blend uniformity, we need to follow the STP only. For relative substance are structurally related to drug substance, and these substances may be identified or sometimes unidentified degradation product, or sometimes we have means under this drug. Impurities arising from the manufacturing process are during the storage of material. So when drugs to be stored for a longer period, the active substance present in it are degraded into some other form. It can be harmful. It can be, sometimes expired batch can have side effects. So for that purpose, we have to have check the relative substances. And here you can see the reagent acetonitrile, perchloric acid, sodium perchlorate monohydrate, ammonia solution, and molecular water. The protocol again, this is the generic protocol. We have to prepare the buffer solution. We have to have stability solution, including diluent and standard buffer solution. Standard solutions prepared as per the standard test procedure. Placebo solution is to be prepared. Sample solution is to be prepared after. This contain different step for different strength of tablet because it is specific for every drug or every tablet. So this cannot be shown here. This is as per the standard procedure. Whatever it is, like you are going for paracetamol, you are going for ranitidine, etc. Then you have to follow that STP only. Under these conditions, column temperature, mobile phase, flow rate, detector, injection volume, front time we have to fix. But these are all already stated in STP. We have to follow because for the new drug, we have to fix. But for the drug which is already in the market, we have completed R&D. For those drug, we have to, because that is all the things has been prepared in STP, in the form of STP, standard test procedure. We need to follow that. Placebo and sample solution are injected into the chromatograph. Then after running HPLC or whatever is required, like sometimes we are going for gas chromatography, then chromatograms can be recorded. Placebo chromatogram for any extra peaks should be examined, should be compared with corresponding peak, and then you can reach on some inferences, some prediction that this can be degraded or not. Other test is disintegration. Disintegration is the breaking down something into the smaller pieces. Small means they have some another constituents. So it can define as a state in which no residue of the unit under test remains on the screen of the apparatus. So it consists of fragment of disintegrated part of the tablet component, like insoluble coating of tablet or capsule shell or any melted fatty substance. So soluble tablets are coated with some type of, and sometimes they are uncoated, sometimes they have film coated, so they can be go for dissolved. They should be dissolved in the water, giving clear appearance. And soluble tablets disintegrate between 3 minutes, but depending on the time of the dosage form. So generally it dissolved in 3 minutes. When examined by disintegration test for tablet and capsule, using the solution at the ambient temperature of 15 to 25 degrees Celsius, or whatever we are going to fix as per climatic zone, 1 or 2 or 3 or 4, 4A, 4B, whatever it is. So here you can see how we are going to fix. After filling water in this apparatus, we need to put the vessels water in this water bath. Then we have to fill the water into these vessels, water vessels. They should be fixed. The bars with the bars. Then we have to attach the temperature probe to fix the temperature. Then we have to check here in the temperature probe. This is the drain pipe, so that after completing the test, we need to drain the water. Whole water we need to drain out. Is the water heater plug? We need to attach the heater plug to heat the water and to fix the temperature 37 degrees Celsius at whatever required as per STP. Now we have to switch on the machine and enter the temperature whatever is required. Follow the instruction. Now we have to wait till temperature reaches to 37 degrees Celsius, because we have to fix the temperature 37 as per the ambient, because as per the human body, internal temperature 37 degrees Celsius. So we need to fix the temperature. After reaching the temperature, we need to run the test. For running the test, we need to click on one. So now we have to put the tablets. Now we have to wait till time, whatever time we have to fix. These vessels will start moving up and down, because under the stomach all these conditions are happening. So under this temperature only we have to fix, we have to wait for the disintegration test. So now test has been completed, and this instrument will beep a sound. Also, this display that test is over. After test completion, we need to drain out the water. Now the test has been completed. Now this water, whatever we have fixed, fill in the hot water bath. So we need to drain out this. Whole water should be drained out for every test. So now drug release. It is a phenomenon controlled by diffusion or dissolution. Sometimes it is by both. So diffusion controlled drug release is predicted by Higuchi in 1963, and there he has made a Higuchi square of time model. So these by manual we need to follow this type of square model exactly. Drug release can be quantified by the measuring absorbance of the released media using sometimes we are going to a hot plate, or sometimes we are going to a plate reader SPLC. We should also measure the blank nanoparticle. Blank means without drug. Simply we consider one is the blank, one is the standard. So here you can see in this picture, like how drug should release in the stomach, gastric juice. It should come out from the binded particles. So the procedure, you can see here the procedure, the stated volume, which is as per mentioned in the STP, we need to take dissolution media. It should be free from any dissolved air. Then apparatus, we need to use two apparatus. The basket is lowered into the position before the rotation. Another apparatus should be used for the tablet or capsule what is going on. So it should be operated immediately at the speed of rotation specified in the STP only. So here you can see how this.
The drug is dispersed homogeneously throughout the polymer matrix tablet. Drug release from tablets with carbopol polymers or Novion polycarbophils is controlled by drug diffusion through the gel layer that the polymers form in contact with aqueous medium. When the carbopol containing tablets are placed in contact with dissolution medium, the following occurs. The drug in the outside layer of the tablet exposed to the bathing solution is dissolved and then diffuses out of the matrix. The polymer swells fast to form a hydrated matrix layer, hydrogel. Due to the cross-linked nature of the polymers, the hydrogel is not single entangled chains of polymers, as is the case with linear polymers, but discrete microgels made up of many polymer particles in which the drug is dispersed. Carbopol polymers and Novion polycarbophils do not dissolve in aqueous medium, rather they swell. In contrast, linear controlled release polymers such as hydroxypropyl methyl cellulose and hydroxypropyl cellulose are water soluble because they are not chemically cross-linked. The hydrated matrix layer controls water penetration into the non-hydrated core and diffusion of the drug through the hydrated matrix. Unlike linear polymers, carbopol polymers and Novion polycarbophils do not dissolve during the release process.
So there should be some precaution, must be taken some precaution. Like the dissolution apparatus should not be subjected to any vibration, external vibration, agitation during this operation. It should not be shifted from one place to another place during the operation. All the part that may come into the contact with the medium should be inert, and medium must be free of dissolved gases. So the preparation, first we need to go for blend uniformity, then content uniformity, and drug release. Three things are done under the dissolution at 37 degrees Celsius. Time has been should be fixed as per the STP, only 30 minutes, 45 minutes, whatever it is required for the particular drug. And after dissolution, we need to have this type of curve, with the blank, with some other type of hydrogels or particles, drug products. So we need to calculate. We can draw. So here you can see when we are taking one example for ranitidine salt. This is the ranitidine drug, and random salt is there within this ranitidine. On the temperature, we have tested the dissolution at temperature of 25 to 30 degrees Celsius, and these metabolites released from dissolution should be tested with SPLC only. And on the HPLC, chromatogram should be recorded with the parent compound or some metabolite compound, that is a reference compound. So for the determination of water, here the Carl-Fisher method is there. So the apparatus like suitable manual, or we can say we have a auto Carl-Fisher's titrator for determination of water in the tablets in the capsule. So here the Carl-Fisher reagents should be utilized, and every time when we are going to use, they should be standardized. So here you can see some means how we are exactly calculating. This is a calculation by using the reagent only, and reagent is your Carl-Fisher reagent, that is KF reagent. And here the formula after putting into with the confusor reagent, whatever we need to weigh, and after weighing, we need to take volume, sample volume also. Volume of KF reagent utilized for the determination of water. The reaction you can see Rn, where Rn is the base, whatever it is, like whatever we are using, going to use. So by using this means this is the actual reaction which happened during the mixing of confusor reagent. So formula is Vf 100 by W, where V is the volume of KF reagent in ml. F is the water equivalence factor. It is the fixed. We are taking 0.1566 weight per volume, and W is the weight of sample in milligram. Here you can see, like if we take as an exercise, we can calculate how much water contained by the tablets. We have taken 3 samples, 1, 2, 3, because we need to take average from the sample selected, 3 samples, 5, 20 samples, whatever it is. So KF reagent for this tablet to absorb 30 ml is required. Average weight of the sample is 650 milligram. And the total strength of tablet is 20. Out of 20, we are taking 3 tablets. So here you can see, by using the above formula, we are going to use water content percentage. So here 30 ml KF reagent into F is 0.1566 into 100 divided by the weight of the sample, that is 650. So we need to find the percentage will be 0.72. In the same means second tablet we are taking from 20 tablet. Now again we require 35 ml of KF reagent. Then again we have to calculate by using the same formula. Then here you can see 0.84% water content. In the third, we have taken third tablet out of 20 tablet. Here 40 ml of KF reagent is required. Then we can calculate the water percentage. Here it is 0.96. Now, what is the acceptance criteria? Acceptance criteria is less than 100%. Oh, sorry, less than 1%. And here we have tested for 3. One have 0.72, second has 0.84, and third has 0.96. So average of all these 3 is 0.84. So that means whatever the sample we have taken from the batch, it is on acceptable criteria. So we can go for it. So we have tested for ranitidine or rhinone salt. So ranitidine tablets are accepted on the basis of water content. Okay. So for friability, it is actually expressed as a loss of weight, and it should be again calculated as a percentage of initial mass. So test of friability is provided to determine the compliances with the requirement which is specified in the STP or individual monograph. Friability of internal tablet is the phenomenon where by tablet surface are damaged or so evidence of lamination or breakage when subjected to mechanical or attrition. So the instrument is designed in such a way to evaluate the ability of compressed uncoated tablet to withstand the abrasion in packaging handling and during the transportation. So what the principle exactly? This is it should be compressed in such a way that should not break during handling, packaging, or transportation. So the test apparatus should be like here. The diameter of this apparatus varies from 283 to 291 millimeter. The depth of this apparatus should be 36 to 40 millimeter, and it should be transparent. It should be made of transparent synthetic polymer. It should be polished with the internal surface subjected to static buildup. And here one side of drum is removable, where you have to put the tablets. Tablets are timoid, oval shape sometimes. So we have to put inside that radius. There should be a curved projection. Here, this curved projection between 75.5 millimeter to 85.5 millimeter that extend from the middle of the drum to the outer wall where we need to put the tablets. And drum is attached to a horizontal axis of the device that rotate on at 25 plus y minus 1 mm. And at each turn, the tablet roll, we can slide, fall about 130 millimeter onto a drum wall, and other side also. Here you can see this is the test apparatus, digital fibrillary test apparatus. Here we can check in digital form, digital panel is there. So how this test should be carried out, here you can see we have to take that tablet, and tablets are here we are taking, and we need to weigh the initial weight of the tablet. We need to write on some piece of paper that how much weight of those tablet initially when we have taken, and after running the test under the fibrillary test apparatus, again we need to check the weight, and then we need to find out the difference between these two, initial and final. So this is the test apparatus drum where we need to put the tablets, and we need to run at here. This is the Roche fibrillator. We have to clean before we use. 10 tablets we are putting here, and this on this drum you can see this is the one projection, curved projection, and after putting we need to click the start button. After start button, this will start 25 RPM. We need to fix. Because this may vary depending on the tablet or the salt, so the strength. So here you can see this is going on, and after completion of total cycle, we need to weigh the tablets, calculate the weight, and also we need to find the difference of initial and final. Initial when we have taken the sample, final after running the test apparatus. So we need to find the difference. After finding difference, one can easily count, calculate that during the transportation, during the storage condition also, how much weight can be lost. Here, you can take your tablets. You should clean the tablet from any type of So again, we have to take the weight of 10 tablets, record the weight. So after recording the weight, we can find the difference between the two weights. So this is the formula, initial weight and final weight. We have to calculate on this basis. So procedure, I think you have already learned about this, how we have to go for either 10 tablet or 20. Here we have taken 10 tablets only of 650 mg. So we can calculate, we have to find out initial weight of the tablet minus final weight of the tablet. If we go for average, like 10 times, 20 times we have done, then we have to find the average. The same way, we have to go for 20 tablets, 10 tablets, 100 tablets even. Now, dissolution means whatever we have discussed in the previous slide, previously, like when we are going for dissolution, the water, artificial gastric juice, or sometimes we are using artificial enteric juice. These are the liquid media. When we are putting the tablet into this media, we need to determine the dissolution rate. And this dissolution rate indicates the potential availability of drug substance for absorption, and also the physiological ability of drug we need to determine. So the instrument you can see here, this is the instrument, means how this is working exactly. So the temperature we need to fix as per the body temperature, 37 point plus minus 0.5 degrees Celsius throughout the operation. And here the means you can see in this picture, the shaft is there to rotate, because this should be rotated. Means we need to create the condition whatever we are having inside our body. Here you can see, for example,
or solid interface, temperature, and solvent composition. There are various uses of the dissolution apparatus in pharmaceutical industry. First, the drug dissolution testing is routinely used to provide critical in vitro drug release information for quality control purposes. Second, formulation and optimization decisions during product development. Third, equivalence decision during the generic product development. Fourth, product compliance and release decision during routine manufacturing. So now let's see how the instrument works. First of all, switch on the mains to start this equipment. After that, switch on this temperature regulating device to maintain the temperature 37 plus or minus 0.5 degrees Celsius. A display screen will turn on, which will show the essential setting data. Adjacent to it, there is a control panel having the set of buttons used to control the system. It is a 8-pedal station system. This is the water holding system in which we fill the water up to the pump which is shown on the apparatus. There is a heating rod which is dipped into the water to maintain the temperature. After that, we have our graduated basket in which we fill the dissolution media as specified. Today, we will study about the dissolution profile of this paracetamol tablet. First of all, we put our paracetamol tablet in our given specified dissolution media. Immediately after, we will lower the platform by the platform down. After the certain level, we will start the stirrer on and further set the RPM according to given the sounds. Now, we will stop the stirrer and withdraw the sample according to the time specified in the model. The same amount of the withdrawal sample will be added by the dissolution media to maintain the same condition. Again, start the stirrer and do it the same procedure till the last one. After the last endpoint, stop the stirrer and lift the platform by the pressing the platform button.
so in this case, you learn about how dissolution test apparatus will be run and what condition we need to maintain, right? And this would be means media, whatever we are going to use, it should be free from dissolved air, and it's the material made of means what all the apparatus should be made of the inert material with where it should be contacted. So here you can see this is the glass only. Some precaution, it should not be subjected to any external vibration. Medium should inert, medium should be free from dissolved gases, and if powder filled with a capsule sufficiently hygroscopic, then it should be stored. It may pull out in the moisture, so it should be granulated. And also we need to use some buffers, gastric juice or enteric juice that should be artificial. It should be free from the disorder cases. pH value, we have the digital temperature. So the digital pH meter, sorry, we have already shown in the previous video that how one can easily find out the pH. Divisibility test, this should be, no, this test should done to verify the content uniformity, that is CU in the tablet. So for doing this experiment, weigh the intact tablets, then break the tablet into the, from the mark line, a break line, then we have to weigh individually, means in the parts. If there is a only one mark, one break line, so there will be two parts. We need to weigh the individual part, and this test should process this test should done for 20 tablets, 10 tablets from the sample. Then what is the variation? Because the whole weight of part 1 and part 2 should be equal to the whole tablet. If there is a variability, there is a difference, then means we need to calculate on the basis of acceptance criteria, and acceptance criteria is less than 1%. For storing the material product, it should be in closed container. It should be protection from the light. It should be stored in the cool place by addition of some, because sometimes the product comes with some other type of substances. These are the recommended condition as per IP. Cold place is 2 to 8 degrees Celsius, cool place 8 to 25 degrees Celsius as per IP, and 8 to 15 as per USP. And ambient temperature, we can say the room temperature 25 degrees Celsius, warm condition is 30 to 40, excessive heat is more than 40, control room temperature is 40 to 25, freezer recommended is -20 to -10. For the stability study, we have studied various environmental factors during the transportation, during the means all the conditions, whatever we have, for temperature, light, intensity, humidity, pH, etc. Because these factors may affect the drug at which they were stored. To minimize the efficacy of drug before its actual expiry or shelf life, they should be stored at a prescribed environmental condition by the manufacturer. The drugs should be stored at having 25 to 30 degrees Celsius temperature and 60 to 70% of humidity in the atmosphere for a longer duration. And the 37 degrees Celsius temperature, drugs should easily dissolve in the gastric juice. We need to study, we need to find out from the stability study only. Minimum time of dissolution should be in the gastric juice is 30 to 45 minutes, or depending on the salt. Environmental condition for the tablet and capsule can be tested before release into the market for a given period, and stability of drug product can be defined. Stability should be acceptance criteria like less than 1%. Water contents again less than 1%. Blend uniformity, content uniformity, it should be checked before going to release to the market. It should be following acceptance criteria. It is less than 1%. So these are the things about stability. Now we'll discuss about the GMP and GWP. GWP is a good warehousing practices. So why it is required? Because as we seen in the stability study, we need to fix the warehousing condition, or we can say storage condition. So the sourcing and distribution pharmaceutical material and the product, which are critical activity in any integrated pharmaceutical supply chain. This involve packaging material, active pharmaceutical ingredient, excipients, and final products such as pharmaceutical or medical devices are the combination of the product at some place that is known as warehouse. So you can see example of warehouse, the picture shown below. So there's some different type of warehouse we have, but we need to maintain the illumination that is known as light intensity, the temperature, and also as well the humidity, because these are the major factor when we are storing the drug in a warehouse. for a longer period, because immediately after manufacturing, it cannot be used by the end user like we. It should be stored, it should be transported to the vendor, then it can go to the distributor, then it goes to the dispensary, then it goes to the medical stores or something. You have to purchase from a medical store or dispensary, then you can use that drug product. So with the globalization of pharmaceutical industry, individual and the organization from different locations around the world are responsible for handling, storage, distribution of these pharmaceutical products. So the warehouse play a very pivotal role, a very important role in manufacturing quality product, as because they are responsible for incoming goods and releasing the final product, a finished good. It is critical to have adequate control over the entire supply chain, ranging from the material procurement to manufacturing and to the delivery of the final product to the patient or to the distributors. So there are two things are there, GWP and GDP. GWP is the warehousing practices, where GDP is the distribution of the product, not distribution only, transportation and distribution. So good distribution practices are there. This specially based means there's only one is storing and transportation. Two things are there. So objective of this warehousing practice GWP to maintain the quality of the product by every organization who are involved in supply chain. They should understand, they should comply with the applicable requirement, and also they should ensure that each activity in the distribution of a product carried out according to the principle of GWP or GDP, that is good distribution practices. And this under GMP, protect medicine from the damage during the storage and transportation, because this enables to the from the warehouse to the means to the dispensary. We need to protect the medicine from damaging, and also GMP play a very significant role to prevent the degradation of product by exposure to adverse temperature condition. It should be avoided with mix-up of the or some contamination by the material. It should maintain product identity, traceability. It also prevent the time expired or damaged material product from being used by someone or distributed in the market. So when the material coming from the warehouse, that is sometimes we are having non-GMP material like non-production consumable, like take example of toilet paper, stationery, etc. Sometimes we have finished good with production material consumed in processing like filter, lubricates, etc. Laboratory reagent is also there. It is coming to the warehouse like buffers, chemical, reagents, etc. And this GMP applies to manufacture starting material, chemicals, packaging component like blister pack, film, bottles, caps, vials, seals, and also it applicable to printed matter. And printed matter can be labels, cartoons, inserts, leaflets, pre-printed with tubes, we fixed with the tubes, etc. Barcodes, everything. So here you can see how warehouse to be made.
Operations. Pharmaceutical manufacturers specialize in research and development, production, sales, and marketing. Delivering these vital products to their final destination is a complex process. Proper handling, storage, and distribution procedures are strictly regulated by federal, state, and other government and regulatory authorities. These stringent requirements make it more economical for our clients to rely on WDSRX for their end-to-end inventory management and reporting. WDS requirements will be implemented prior to their scheduled deadlines. With over 210,000 square feet available for order receipt, storage, and fulfillment, WDSRX provides multiple diversified services performed to the highest quality standards, including inventory and data management, transportation management, order-to-invoice processing, and temperature-regulated handling. Our facilities are protected by multiple and redundant systems to ensure inventory security and data integrity. WDSRX
So these are the condition requirements. And what are the procedure It should be arrived from a supplier. It should not be damaged. These are the responsibility to check. And these means drug products are correctly labeled and identified with supplier's lot number for traceability purposes. It should means they should be given a unique in-house lot number. Every material coming into warehouse should be registered in inventory, that should quarantined, then sampled for release testing. Some consideration in layout of the warehouse of any typical warehouse. It should have quarantine area for storing goods that are not yet been we can say tested or inspected. Material stored in quarantine cannot be used for release until approved by quality control. And a warehouse should have dangerous goods storage area, separate dangerous goods storage area, and to ensure the safety of the staff and the facility, like flammable goods such as ethanol, it should be stored in a dangerous goods storage area. Specific storage condition like lime-proof cabinet should be used here. Warehouse should have area restricted goods like poisons, drug of addiction, etc. So, and there should be restricted to some general staff. They only specifically authorized staff should enter into that area. Another one area should be isolated area. A separate area should provide for faulty and recalled goods. To ensure that they should not be sold by mistake or should not be issued into the market. Another area, reject area. It should be reserved for rejected batch or expired batch something. So we have to go for this. And cool room is usually operated to 2 degrees Celsius to 8 degrees Celsius of the temperature. Here, the vitamins or sensitive materials should be stored in this condition only. Good housekeeping. If we are maintaining good housekeeping, that means everything in its place. So it minimizes the opportunity for mix up of the goods, and also it reduces the possibility for dispatch or issue of incorrect goods and improve the warehouse safety. So the good housekeeping at any warehouse is a key to successfully operate a warehouse. Generally, warehouses are very busy with a constant movement of inventory in and out, as well as a different warehouse location internally. So also this warehouse protecting the receiving area from the weather. Here, the checking regularly to ensure the containers, packs, or package, it should be properly sealed and labeled. Cleaning facilities should be done regularly, and cleaning up spills immediately after happens something. And for the control of the product label in the warehouse only, one should means everyone take different responsibilities. So each different type of label is clearly identified and segregated in a label store to prevent the mix-up. Access to the label store must be restricted to authorized personnel. There should not be loose approved or leased label in the store. Any unused or damaged labels should be returned to the quality department. If there is approved or released label, they should be used only, and which is released by the quality department, QAQC. Before release of the labels, they must be checked and double checked against the work order to verify the product name, their code, label identity, version number, coded batch number, and also the expiry date. The barcode, or sometimes we can say material code. It looks like that. Here you can see on the slide. So material item is linked to the standard name. It is a master batch record, MBR. Also, it can be used in formulation. This code corresponds to the barcode, usually help to identify approved supplier or grade of material. Some companies apply a unique barcode, providing a second unique identifier for a material and as well the product. So the barcode is usually it is used electronically scanned to identify the item before use. Sometimes the these warehouse are using UIN, unique identifying number. This is sometimes it is known as GIN, right? GIN means goods inward number. Or sometimes this is also known as lot number, a batch number. So these are the same one, UIN, GIN, lot number, batch number. These number is critical because uniquely identifies and allow traceability of the consignment. So it is recorded in the laboratory on the computer system and in production when product is to be formulated. Storage condition in the warehouse, like whatever the material we have received or the material to be stored for release, this should not be degraded. And if can degrade, if not stored under the approved storage condition. So we warehouse should follow the GMP, GWP, CDP. And all the storage conditions should be registered at the government, making it a legal requirement to store them in a compliances. Here you can see the handling and storage SOP. These means this SOP has these major 5 points. Objective is to lay down a procedure for proper identification, storage, and handling of raw material. And the scope is SOP shall be applicable for storage and handling of raw material packaging material. Responsibility is the store assistant. Accountability is the head of department. Procedure, like you can see one by one. After receiving the material, store person shall unload the material in the staging area and clean the material with the help of vacuum cleaner. Store person shall also record his observation in a physical verification. We can see the arm physical verification record for PM. When the material should be released by the QA/QC person, so it will be fixed with approved label. For the storing of raw material, it should be stored at prescribed condition, whatever it is written on the label, like 2 to 8 degrees Celsius or 25 degrees Celsius. So this should be released by QC. If it is, material rejected by QC department, then it must be shifted to rejected storage area. A logbook of rejected material should be maintained. Information of the rejection of the material should be given to the purchasing department. After dispensing, the loose quantity of material should be closed tightly with nylon thread. In case of temperature box store exceed the limit, information to maintain, so it can be rectified. Rejected raw material shall be sent back to the supplier. If rejected material is destroyed in factory premises, then prepare the destruction note and again follow the SOP, like packaging, printed labels. After QC department, they approve the label, then approved labels should be used for labeling on the material or boxes. And for the any type of plain aluminum foil, PVC, PVDF film, so all these things should be utilized after QC approval. Measuring the plastic jar, container, cap, everything it should be utilized after QC approval. And keep small consignment or leftover quantity on the racks or pallets. Issue all packaging material as per the basis of the production department. It is mentioned in the SOP. So this is the exactly instrument. What type of instrument in the warehouse? Case trolley, drum carrier, drum lifter, drum tilter, weighing balance, hydraulic trolley, and dustbin, writing table, stacker charger, standard weigh box, vacuum cleaner. These are things are required. For receiving the material into the warehouse and to dispatch the product, finished goods from the warehouse, this type of system to be followed. Here is the storage, or we can say the warehouse. On the basis of use or issue, we can release because this warehouse is intended for 2 type of product. One is the raw material, another is finished product. So it can issue or they can also, they are using some type of material. So if any article, machinery, equipment, instrument is not working properly, then it goes for the maintenance. If the life some expired medicines are there, machinery is not working, then it should be going for disposal. But everything, whatever is going from the warehouse, it should be informed to the accounting for database preparation. One. Second one, for receiving the material, it should again coming from the accounting or purchasing department. From it should be stored in database. Demand should given by the department, different department, from the laboratory, from the testing laboratory or something. Then procurement should be done on the receipt. It should be stored in the warehouse. So means receiving and dispatching from the warehouse only. This is two-way process. So one material to be dispatched as a finished good, and the material as a raw material which is utilized by the manufacturing facility or laboratory. Basic principle here you can see. Hursley has identified 7 basic principles that should be followed during the transportation or in case of warehouse management practices. It should be effective management, supervision, sound purchasing method, efficient purchase system, and also it should have a simple inventory control program so that one can easily understand. Result-oriented acquisition and distribution should be there. Written policy, procedure, whatever the policy you have, you should write down with a quality management system, and a practical receiving program should be there. Once the list of drug had been finalized, means there is a demand has come from the laboratory, it should be estimated on the basis of consumption pattern during the last 2 or 3 years, and the objective of district health officer are including hospitals, dispensary in the vicinity. We can also estimate how much quantity of drug is required, and we can go for cyclic changes, or we can say go for some type of epidemiological occurrences of the disease, like coronavirus or corona pandemic. So these type of things are available to estimate the drug demand of the drug, a particular demand of drug. During the procurement exactly the process of acquiring supplies or something. 3 major sources. It can come from purchase, come from donation, it come from the dispensary, district health center, hospital. So it depends on the demand. The cycle of the procurement. Procurement is a cyclic process which requires a sequence of steps to follow. And also here there is a timetable for everything to procure from the market should be clearly specified and closely followed. These are the steps. Review drug selection by drug selection committee. Determine the quantity of drug to be required or purchased. Reconcile needs and funds required for the drug procurement. Choose procurement method, whatever method you are going to apply. Select the suppliers or vendors. It should be registered with the company or organization. Specify the contract term, MOU. Legal aspects of the contract should be framed, term of reference. Monitor order status in the essential, because this is the monitoring stage. Then receive and inspect the drugs supplied by the selected vendor. So acquiring means during the purchase system or procurement, it requires suppliers as because this should be having optimum cost, and you have to go to the registered vendor only to obtain supply in a high quantity, and also they should assure prompt delivery or dependable delivery. They should distribute the procurement workload to a period of idleness and overwork. For that reason, we need to optimize the inventory, and we need to have a system in the warehouse where we are going to store the drugs. Storage material are received at a central place in the receiving system section, and loading and loading supply, like we can say a number of containers of consignment or unit loaded or loaded by the supplier. It should be checked during the unloading. The number of containers or consignment unit on the freight bill should be compared. It should be checked. And A and B, it should be checked, like how many container has been sent by the supplier, how many containers are received during unloading. Then inspection of the container consignment or unit for any external visible damages. If any damages, then you can reject. Notifying the supplier regarding any discrepancy or any issues, whatever we have mentioned under A, B, C, or D. If it is there, then you have to just inform to the supplier. For unpacking, verification of the material received from the supplier according to the packing slip. Verification of the quantity of supplies, inspection of general condition of the material received, like broken or leakage or date of expiry or substandards are there, then immediately you have to inform to the supplier. Another one is the note, goods received note. Copies of the completion of received report or goods received note is sent to the supplier, and also it should be ultimate user. It should have that item, the quantity received, and item and quantity on purchase order not received. If it is not received, whatever is purchased or order in the work order, then we have to inform. We have to mention under the goods received note. So entire material management trust may be built on managerial function of the planning, organizing, directing, controlling, and staffing. So in case of sound packaging method, this is actually means ethical, and it has a hard poised negotiation. So in case of purchasing from the district healthcare organization or from the hospital or by the hospital, etc. So it depending on the type of situations and negotiation for better prices, the supplies. Monitoring of the expiry drugs or drug product. First, drug products should be purchased on the basis of requirement on the quarterly basis instead of bulk or for whole year or 2 year. We need to purchase the drugs on the basis of requirement. While receiving drug, it should be ensured that there is sufficiently long span time to consume it before the expiry date. And this should also be an agreement with the supplier to replace the drug expired by fresh supply. It shows a simple way of keeping check on expiry date of drug. A hospital, PSC, they can also maintain a separate register for expiry date. As the drug are received in the store, they should also enter a chart, make a chart. This chart should have date of expiry. If your drug is going to expire in September, in February or something in a particular year, then it has been listed. How you are going to list? Here, two things are there. Three things are. A sign of X, that is cross, is entered in the month of expiry against each drug. Like if your drug is to be expired in the month of February only, then when you are going to inspect, you have seen that your drug is to be expired in this month only, then you have to put as X. If approaching to the expiry date, then you have to put O dot. O dot indicate the approaching expiry date to the next 6 months. Or we can say next before more than 6 months, not 6 months. If approaching less than 6 months, or we can say after 6 months in the month of expiry, then O dot can be replaced with the X. So storekeepers should be trained to take necessary action for utilization of this drug as a drug reaches sign O. If it has O, that means it is approaching to expiry. So it should be utilized before expiry of that. And for consumers consuming within the system, within the manufacturing or in the hospital, either as a raw material, either as a means your product. So we have to use a double shelf system, a 2-bin system. This kind of no paperwork system of the stock control. Stock of each item is physically separated into 2 bins, working bin and reserved bin. When the working bin is empty, the storekeeper changes to the second bin. that is reserved exactly. Now reserved bin is working as a walking bin, and it is altered that new supply are needed. Like in every bike or every vehicle, we have a reserved capacity. So in that way, we can work when the reserve bin is to be utilized, then that means there is a requirement of supply to procure. Reserve bin should contain enough stock to cover the expected lead time, depending on the time period, whatever we need, 1 month, 2 months, or 15 days, or some days. So checklist of all item in a stock can be used to indicate in advance the item for which the first bin is empty, and this can be replaced whenever it is required. Quantity in each bin could be established in such a way that each new delivery would be placed in reserve bin and the old reserve stock moved to a working stock bin. Here is the SOP for dispatch. The objective is to lay down a procedure for dispatch, and the scope is means this is applicable to dispatch of the finished goods outside the factory premises. Responsibility is store assistant, accountability of head of department. Procedure here it is like after completion of batch packing, a department shall give information to the BSR. BSR is a bounded store room, and BSR person shall check the following parameter: quantity of product, detail of the labels, quantity in the loose box. So this person, BSR person, shall keep all the finished good in orderly fashion. And after completion of analysis by QA, QC department, verification of batch will be recorded by a person. Finished product release note. Before loading the material, check the following point, like in terms of whatever, like batch number, lot number, etc. We have to check. Prepare excise invoices. Loading needs to be done in the presence of BSR person. After completion of loading, hand over to the duplicate copy to transporter. One copy of each invoice shall be given to the account department. Complete all necessary entries of dispatch in daily stock account register. Thank you very much. Here, whatever as abbreviation we have used, SOP is the standard operating procedure, QA the quality assurance, QC the quality control, STP is the standard test procedure. So now if you have any question, you can ask. Also, we have open platform. You can ask on WhatsApp 990996895. You can call this number. You can send us email. Otherwise, you can also go to this website. Here, one feedback or inquiry form is there. In that also you can mention by mentioning the training name, you can ask your question. We'll get your question. We'll reply immediately as soon as possible. Thanks a lot. Now we have a third more than 45 or 50 now. Now we are reaching to the 50 books in different domain. Even one book on stability studies of drug product. So everything is in place. So now means if you have any doubt, any question, you can ask. You are free to ask anything related to this training session. Thanks a lot. Thank you very much. Now I am ending this session.